Good morning, everybody, or good afternoon, I should say. Welcome to today's seminar. Very pleased to be able to introduce this seminar by Professor Neena Mitter from QAAFI, from the Centre for Horticultural Science. Before we start, I would like to acknowledge the traditional owners and the custodianship of the lands on which we meet today, and I pay respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. To introduce myself, my name is Matthew Morell. I'm the Director of QAAFI and I'll be the facilitator for the seminar today. We have 1 hour allocated for the seminar. At the conclusion of the presentation, we will have a Q&A session. So if you have questions that you'd like answered, please don't pop them in the chat, but please use the Q&A button at the bottom of your screen, and that will fast track them through to us to be able to address them at the conclusion of the seminar. So to introduce Neena, Professor Neena Mitter is the founding director of both the QAAFI Centre for Horticultural Science and the Director for the Australian Research Council Industrial Transformation Research Hub for Sustainable Crop Protection at the University of Queensland. The Centre of Horticultural Science commenced operations in 2018 and constitutes over 70 core staff and students and champions over $37 million worth of research projects, and is committed to developing the horticulture of tomorrow, which will be the topic of much of the presentation today. As director, Neena has overseen 3 major research themes at the centre: horticulture, crop breeding and agronomy, plant protection and emerging technologies. And these operate across multiple regional centre locations and operate in close collaboration with industry growers and government to boost innovation, productivity and economic growth. So Neena's career and passion for delivering real-world outcomes has been recognised with honours, including a fellowship to the Australian Academy of Technology and Engineering, a Young Scientist Award from the Prime Minister of India, Queensland International Fellowship and a Gates Grand Challenges Explorations Award, and Women in Technology Outstanding Life Sciences Award. And Neena is driven by a realisation of agriculture's role in shaping the world economically, socially, environmentally, and politically. Now, many of you will be aware that Neena has taken a career choice to move on to a new role as Deputy Vice-Chancellor Associate at Charles Sturt University, and indeed yesterday was Neena's official farewell at UQ, and Professor Deborah Terry and other illustrious guests were on hand to celebrate Neena's contributions and to wish her all the best in the future. And I think one feature of that, aside from that event, aside from the focus on the research achievements, industry engagement, collaboration, was a real recognition of the passion that Neena has had for developing early mid-career researchers and students, and her role in promoting equity and diversity. So many great aspects of Neena's contribution fully celebrated yesterday. More of a focus on the science today. So on that note, I would like to Again, congratulate Neena on her contributions. Wish her all the best for the future. But hand over to you now, Neena, for your seminar.
Oh, thank you, Matthew, for that warm and kind introduction. And really pleased to deliver this seminar, kind of an exit seminar, but really looking forward to it. As I always say, we are going to keep connected and keep delivering together. So without much ado, maybe I'll just start my talk. Thanks everyone who have joined online now at the time slot, and thanks to those as well who might watch this presentation later if they can't join today. Change and innovation. When we start thinking about change, sometimes the change can be acute and we feel it. At times, change can be just a continuum. It keeps on happening, and we feel it only when there is a jolt. Now, it can be climate change, geopolitical situation change, market access issues, trade imperatives, regulatory change, or even just a change on how we think about food, how we perceive food, taste food, how we look at its color. All that actually presents to us researchers both challenges, but as well as opportunities to innovate, and innovate in a way that transcends the boundaries of geographies and disciplines. So that is where I will try to give you a little bit of taste and flavor of what we have been doing at the Centre for Horticultural Science in QAAFI. But I'll start with maybe me crossing over the border. So I crossed over the border from India to Australia in the year 2000, of course, motivated and passionate to address some of the global challenges, and landed in this most wonderful University of Queensland. And then in 2014, became part of QAAFI when it was starting. And as Matthew mentioned, we formed the Centre of Horticultural Science in 2018. And my focus today will be on the horticultural science. So crossing of borders started. And of course, when we talk about horticulture, food and nutritional security, think about it, fruits and vegetables are nuts, fruits and vegetables are really centerpiece to it. You know, FAO recommends 400 grams of fruits and vegetables per day. But then there are issues of non-availability. More so, this is constantly increasing now. When I travel to different countries, the conversations have changed. People talk more about fruit, nuts, and vegetables than grains, just because of the issues linked to the nutrition. And that's reflected in a change in the horticulture market as well. It is really seeing a global growth, and this is what the projection is, the CAGR of 9.9% as we go from 2022 to 2031. And this is happening at home as well. So that was the global figure. If we look at the Australian horticultural production, this is a report just released last week or in August, early August, gross value of Australian annual horticultural production, 18 billion projection. Includes, fruits and nuts, vegetables, nurseries, and other horticultural elements as well. But key to it is that we are also very much focused now on exports as well. Whether it is imports and exports, borders are important. We really need to protect our borders, and we need to see and innovate. How do we cross those borders when we are thinking of either importing the horticultural produce or sending it to overseas? Another thing that is changing is when you see these market projections, you not only just now think about productivity market projections or market rate, you have now websites. What is happening to horticultural lighting space? What's happening to biochar market in horticultural space? What is happening to water treatment, packaging? And this list was too long when I was going on that website, once again showing that there is a space to innovate across different things that now constitute horticulture or can deliver into what we think about horticulture. And when we think about change, I just found this old one in 1995, a presidential address where Daniel Camcliffe says, he's convinced that at no longer other time in history have changes been more rapid and challenges been greater than at present. So there is always that question of change and challenges. And in 2017, when we were forming this Centre of Horticultural Science, came across this from Fred Steele. truth is, the face of horticulture is always changing. Like climate change, we don't notice the impact until it directly affects us. So, and this picture once again summarizes some of that. This is a packing house in California, 1905. You can see only 12 women working in the packing house. Fast forward, state-of-the-art Hallows packing house, size of 11 football fields, completely automated, no human being inside. So innovation is driving and delivering into the horticulture of tomorrow. And when I think about borderless, as I mentioned, transacting disciplines and geographies is the cornerstone of addressing the challenges of food and nutritional security now and into the future. Whether it is innovations in breeding or precision agriculture or biotech, whether we look at that holistic interdisciplinary approach, one that integrates not only science, technology, sustainability, social science to meet the needs of a growing population. So that is where I bring in that borderless innovation, not only enhances productivity, but fosters resilience in the face of unpredictable climate and market shifts. That brings me to our wonderful Centre for Horticultural Science, founded, as I said, in 2018. And we set the mandate even then, solve tomorrow's horticultural challenges together today. And I thought it was apt for me maybe to show you a brief video that we made, because it does capture some of the spirits of this center.
A vibrant and growing horticulture industry is required if we're to provide nutritional food for 10 billion people by 2050. Horticultural products like vegetables, fruit and nuts are vital for our health. Horticulture offers growers a high-value, high-return product with export opportunities to Asia and globally. As consumer demand for horticultural products continues to grow, the Queensland Alliance for Agriculture and Food Innovation, or QAAFI, at the University of Queensland has established a dedicated Centre for Horticultural Science to build on QAAFI's world-leading research expertise to support horticultural industries in tropical and subtropical regions of Australia. Our researchers have a global reputation for innovation in crop improvement and crop protection. QAAFI partners with government and industry globally to meet environmental challenges and manage pests and diseases. QAAFI offers an integrated research capability to help develop new products in industries across the horticulture, broadacre crops, livestock and food and nutrition sciences. Our researchers are committed to delivering innovative solutions to grow horticulture in Australia and internationally.
So yeah, this is sort of resonates with me every time I watch it. I feel there are elements that we are continuing to grow as we grow this Centre for Horticultural Science at QAAFI, not only just as a standalone center, but integrating it with QAAFI's other centers, integrating it with the University of Queensland, and of course, very key to it is our collaboration with DAF, strengthening the spirit of true alliance, I always call it. We are really getting the best of both worlds through this alliance, forming the Centre for Horticultural Science and broadly in QAAFI. If we look at our program areas, which we keep on sort of wordsmithing a little bit to reflect that change, but pretty much that's where the innovation stands, whether it is advanced production systems or genetics, crop protection, smart propagation. But key to that innovation is also about training and capacity building. There can be innovations in how do you deliver a course, how do you do a training, or how do you excite an HDR to work with the industry or motivate them to become key players in the horticulture sector. I'll just give you some glimpse of what we have been doing. So this is just one of the new program areas that we have added in the Horticultural Centre just about 2.5 years ago. So I thought I'll start with that, led by Professor Paul Gauthier here. We are looking at indoor farming, whether it is greenhouse horticulture or it is protected roofs or it is completely indoors. This area is growing at an unprecedented rate. We do have to address the challenges of energy costs, but there are huge gains to be made once we really address that return on investment issue, but also pretty much get geared up to address the issue of climate change. Genomes, genetics, phenomes, traits, and predictions, of course, genomics is really turbocharging genetics and huge opportunities. Hort Innovation funded projects, which we have at the CHS in partnership with other universities and collaborators, once again, crossing the borders of state borders and national and international borders, and looking at different crops that are of relevance to tropical and subtropical horticulture. Very recently, just maybe a month ago, we have now been become a part of a $41 million Hort Frontiers program. Once again, the reason for showing this slide is, look at the partners. It's led by QT. There is number of partners involved. There's industry involved. There's international partners involved. And the key traits that we are looking at just resonate with what does a next generation orchard will look like. We are also working on developing some low-cost method for genotyping by sequencing. So this is where new advancement in sequencing comes into the picture. There is nanopore skim sequencing. And some of the lessons here we have learned from the use of this technology in our Centre for Animal Science as well, where they were working on beef genomics and low-cost genotyping there. So these are lessons then translates into our module in horticulture, where Associate Professor Craig Hardner is working on this project and also looking at some new genetics for the emerging industries. Anything we do in genetics, genomics space, then the realization of the impact comes from some of the major breeding programs. And here is a macadamia breeding program where the center has released 4 new varieties in collaboration with DAF, and are continuing this program, looking at thinner shells or bigger kernels or tougher husks or pest resistance to deliver much more impact. But where the transaction of transdisciplinary technologies now come is Mobeshwar Alam now looking at, can we use artificial intelligence in the improvement of prediction accuracy? For my age group, artificial intelligence presents a lot of challenges. So I do struggle when I think about artificial intelligence and how do I use it. But this is a space which is growing in every aspect. When we think about AI and machine learning is becoming a part of it. And we have joined this and we are looking at it. How do we do the genomic selection? How do we shorten the cycle? And how do we that improvement of prediction with accuracy? We also, as I was mentioning, we go work across centers in QAAFI, across UQ, along with a large number of industry partners. We now have an ARC training center in predictive breeding with university, industry, and government partners. And once again, if you look at the new technologies section of this breeding center led by Lee Hickey, there is artificial intelligence, there's modeling, there's multi-omics, there's high throughput phenotyping. So yes, this transdisciplinary crossing of the boundaries is very much essential. And you can see the center is about animals, about grains, about chickpeas and cotton, but also about apples, grape wine, macadamia, strawberries. So this is where those boundaries become fluid. And as I was mentioning, it's about genetics, but it's also about taste, perception. That's another border that we have to cross. Professor Heather Smythe at the QAAFI Centre for Food Science is really looking at consumer preferences, sensory profiling, linked to genetics. And this is what is very important when we look at new horticultural products with high consumer value. We are doing physiology and modeling. How do we predict what the next orchard should be in terms of whether it is going to be optimal in terms of receiving the light and other features. So what it should be, and that's being done through modeling. We are also looking at digital space, twins, and once again, AI. So innovation of orchard systems, because once you plant an orchard, it's pretty much you're done. You need to now wait for 10, 15, 20, 30 years, and it's going to be productive for you. You can't just replant it next year or change the topography next year. And therefore, we have projects which are looking at how do we design the next orchard? How do we do the physical twin and the digital twin to advise the growers of what would be the best row spacing or best layout for the orchard? And also another element of that digital twins, along with DAF and industry partners, is looking at spray practices. It's really important that whatever we are spraying on the orchard should reach the tree rather than falling onto the ground. And as you can see here in this slide, it really shows just by simply changing, doing a digital twin modeling, simply changing the angle of the fan or the flying height, we can make most of the thing fall onto the canopy. We can really have that saving rather than having it missing the canopies. So most of the spray is now hitting the canopies, and this could be very important in terms of use of pesticides as well. We have the diagnostics programs that run, once again, very globally connected, because this is important when we are either crossing the borders or, as I said, we are bringing something inside. We had a big program on banana bunchy top, viral pandemic. Once again, this is about not only just protecting our borders, but helping others who are facing this challenge of this deadly banana bunchy top virus. Avocado biosecurity, once again, sorry, I'll jump the slide. Here is an exciting stuff I just wanted to mention. So Andrew Gearing here in the center is actually using bee-assisted surveillance of avocado sun blotch viroids. So see how the innovation really progresses from one thing to another. Bees come to pollinate these avocado trees, and therefore the detection of the viroid in the bees can give us an indication whether the disease is present in the orchard. Similarly, in the macadamia space, there's a huge work going on in terms by Femi Akinasami on modeling, forecasting, looking at epidemiology, looking at diagnostics. So no boundaries across that, just that whole picture of an impact being delivered in the tree nut protection. I'll now come to two areas of my own research area and dwell a little bit more on that as my last seminar here. This is once again resonating with crop protection. We all understand the issues with pesticides. The European Green Deal, asking for 50% reduction in pesticide use. You can look at the stats. I'm sure people listening are aware of the issues as well, whether it is emissions or runoff to our precious waterways or lack of specificity. We do not want to harm our beautiful butterflies and moths, or the issue of residues. This is where an innovative space of using RNA-based crop protection comes into picture. RNA interference, and I won't go into the details of science of this technology, but has been used for generating transgenic or genetically modified plants that could be disease resistant. There are issues, of course, regulatory issues or acceptance issues, but more than that, for me, also it was the cost and time involved in generating these GM plants. And it's not very easy when you're thinking of some plants like crops like tomato might be easy, but most of the horticultural crops, we do not even have protocols of how to genetically modify them. So that in itself becomes a long-term question. So the question then we had, the idea or innovation we had was, can we just spray RNA on the plant instead of genetically modifying it? Surprised, as early as 2003, it was shown that it works, as you can see in that leaf picture in a paper by Tanlado et al. At 2003. And I was surprised why this technology hasn't gained momentum. And then you reach the end of the publications. Naked RNA, when you spray on a crop, can be unstable, can get easily washed off, very short protection window. So therefore, not a viable system for growers. And of course, other concerns as well. So the question that we had wanted to answer was, how can we convert these naked dsRNA experiments into a commercially viable system for farmers? And do we need something else? Do we need some carriers to deliver the RNA cargo to make it possible? And this is where the concept of our BioClay was born as RNA-based biopesticides. where double-stranded RNA is the biological active, but it is the clay nanoparticles that act as carriers of that active and provide those features that we were looking at. And you can see on one side, I have listed different technology, different disciplines that have come into picture as we have been developing this BioClay platform. And I might have missed even a couple, whether it is pathology, nanotechnology, bioinformatics, biochemistry, imaging, policy, social licensing, commercialization, all become a part of developing this platform. And we, as Matthew mentioned as well, we do have an Australian Research Council hub, and very pleased because it brings into its fold multiple RDCs. So we have GRDC, Hort Innovation, Vine Australia, and CRDC as partners. So thanks to you, those RDCs, and also all the state government partners, industry, and international partners. The clay, as I mentioned, it's a nanotechnology coming into picture. Clay is just magnesium and iron now that we use. Clay is positively charged, RNA is negatively charged, and it is just a charged binding. BioClay, as I mentioned, positively charged clay, negatively charged RNA. The beauty of this clay is it's benign. It really is biodegradable. I know we use the term biodegradable sometimes loosely, but in this case, it does break down in the presence of moisture in the atmosphere and carbon dioxide into magnesium and iron, leaving no residue. And our aim was to get extended stability to make it viable for growers. For me, it's almost like nature versus nature. RNA from the pest or pathogen to kill the pest or pathogen. And we have come a long way since we started, and it does work. As you can see, we have worked with viruses of capsicum, lettuce, and zucchinis. We have worked with whiteflies as vectors. We have worked on fungal diseases and even gone to tree crops. And I'll give one or two examples here, because this was our first seminal paper that we published in 2017. And if you look at the panel A, you can see that this black blob shows that even after 30 days, we could detect the RNA. So we have extended the stability of the RNA by using BioClay. It does not get washed off. This green color shows it's a fluorescently labeled RNA, so it does not get washed off. It enters into the plants. And most importantly, this picture where we have shown that RNA can actually, we sprayed the leaves on day 1, we challenged it with the virus on day 20, and the leaves were still protected. And this is once again an example where we have of zucchini yellow mosaic virus. This is something which we have taken to kind of a semi-field trial. Took us a little while. Dr. Carl Robinson has been successful in doing this. And as you can see here, it gives us protection. BioClay gives us an extended protection from zucchini yellow mosaic virus. Not only on plants, but post-harvest implications as well. And as you can see here, this is on tomato. And one can see that the tomatoes were sprayed with dsRNA, either naked or as BioClay, and you can see the controls as well. And it really does afford protection. And this is against fungus called botrytis or mold. And this work has been done with the University of California, Riverside. And we are really continuing this work. That team is also looking at not only tomatoes, but post-harvest, such as strawberries, grapes, and other horticultural produce as well. As I mentioned, we came a long way after doing the viral work. We have also done a huge body of work on whiteflies. And I'm just showing this slide as a placeholder here. Happy for people to go and look into this publication. There is a lot of data there, which actually shows that BioClay can provide protection not only against adult whiteflies, but nymphs and eggs as well, and is very specific. It does not affect your stingless bees or even the related pest aphids. So this has been a really major work that we have continued. And this is important because this has shown us that we can target even sap-sucking insects. So whiteflies actually feed on the phloem sap. So we can target sap-sucking insects using this platform. Crossing borders as the ethos of this talk. When we thought of crops, we thought, oh, how about trees? And this is where Anne Sawyer and a PhD student, Beck Degnan, came into the mix. Sorry, my slide seems to be jumping. Yeah, so in this case, we first time showed that we can actually target rust of an important species, lemon myrtle, in Australia. Lemon myrtle, of course, if people don't know, it's really a native species, huge significance to us. And myrtle rust is a big, big issue on that crop. And also, if you can see the image, we were lucky enough to get, the front cover of Molecular Plant Pathology. But what I wanted to show again was, if you look at those orange colored spores, it actually shows what the power of imaging is. So this is where imaging shows us that the RNA can enter into the spores and gives us confidence that this platform is going to work. And this is again showing how it just protects against the myrtle rust after sprays. But another jump that has been made in this myrtle rust scenario is that we have shown it not only prevents, but it even cures infection. So this is the first time that we did this work. So we sprayed myrtle rust infected trees with the RNA, and we could show that the plants recover from this myrtle rust infection. And Sawyer and Beck and the other team members, along with DAF team members, Louis Shoei and others, are really progressing this work now as to how do we translate this to actually make a difference in the forestry and nursery section. We are also looking at technology translation into protected cropping, So protected cropping, we know that we cannot use a number of pesticides in protected cropping, and therefore BioClay platform or RNA-based solutions can present an attractive solution. If people have been reading news, biosecurity risks, Australia has now just detected tomato brown rugose virus. I hope I'm saying the name correct. And that's a huge, huge threat to the protected cropping industry. It really wiped out in some years, entire tomato produce in Belgium and Europe. So we need to be really prepared against such threats. Crossing borders again. So we have crossed borders to deliver into this BioClay platform with India as well, and working very closely with India on things such as whiteflies, thrips, but also viruses like cotton leaf curl virus, which is a biosecurity threat for us, but is present in India, and also looking at viruses of vegetable crops as well. So watch this space as we deliver more results, and once again, crossing borders, working with industries across the borders, and bringing that wealth of knowledge and information that can deliver impact. And regulation becomes a part of this picture as well. As you can see, OGTR, of course, has declared in Australia, it is non-GM. But if you look at the regulatory space across the globe, there is differences. We really do need cohesion. What is the APVMA ruling on this? How does EPA looks at it? What is Canadian landscape of regulation? So we are looking at, as I mentioned about our hub. In the hub, we are looking at the Centre for Policy Futures at UQ is involved in looking at this regulatory landscape, but also on public perceptions. How do we talk about RNA? How do we communicate it to growers? And that is one of the key elements as well, which is looked at by Professor Peter Ashworth, who has now moved to Curtin University. Borders, once again, borderless innovation, absolutely pleased. You can see the number of flags here. Happy for anyone else, if they want their flag on, contact us. We can make you partners in this journey. And it's beautiful to see that we are all delivering together, growing as a big team into this space, and looking at all the way from identification to design to glasshouse to field, but also bringing in elements of scale up as well as social licensing, as I mentioned. And very pleased, so I thought I'll put it in Hub at a glance. It's been 4 years of this hub, but if you look at the delivery impacts, we have 48 plus publications to date and 55 conference presentations, wonderful team of students and EMCRs, all delivering and also learning how to balance the commercial reality with publications. And this is a really big part of their training to understand the commercialization landscape, industry needs, but also then how to balance their career pathways in terms of publications and presentations. So really good innovative space there. And promise of RNA-based biologicals continues to grow. Taken, the dial-up specificity for new targets, ever-decreasing cost of RNA production. And the first product actually has been now released for market use in USA by Greenlight Biosciences, which is Calantha, for targeting the Colorado potato beetle. So watch this space. I believe in terms of bioclay, we do have an Australian-owned and invented innovation, and the possibilities are endless. Again, crossing boundaries, we are going even into looking at animal health, but design of regulation and public opinion are crucial. Just changing gears here, and the nice slide, because yes, I am changing gear to show you another key area where the research is going on at QAAFI, and that's about smart propagation. It all begins with the seed or that beautiful plant, which has the desired characteristics, is disease-free, on-demand clonal supply of plants. But not only that, this tool then gives us the power to do targeted crop improvement, whether it is gene editing or speed breeding or interspecies crossing. It also gives us the power for climate safe storage or germplasm banking, also provides us with opportunities to look at cellular egg proteins or pharma, foods and fibers, if we can ramp up those production scenarios. And my journey into this space actually began when I started working with avocado and fell in love with avocado. Of course, the world's trendiest food. You talk about cartels, mafia, it has every element into it. And the millennials, when we talk about smashed avo on toast, it still does ring a bell. And what the team here at QAAFI has done is we have developed the world's first plant stem cell-based propagation of avocado. That is, we can generate 500 plants from taking a cutting from a good tree, and the cutting can be as small as 0.5 centimeter. This has been, so normally it would take, if you take an avocado cutting from a tree to develop a clonal plant, the process is about 18 months. But having said that, it just gives you one plant per cutting. Here we are getting 500 plants. We can have 25,000 trees sitting in a 20 square meter room in our labs, or a 10 square meter room. And this technology has been commercially licensed, so has progressed, and there is a global outreach and interest. It has gone from lab to field. Once again, the innovation, bringing in the industry partners, addressing the needs, addressing that need for that change, and then making it possible with the help of a good, vibrant team. So yeah, I wanted to show you those beautiful avocados. I'm sure everyone is fond of them. Again, what the new element that we have brought into it, and this comes from the Centre for Crop Science, Associate Professor Sudhir Yadav has been working with us in delivering life cycle analysis. And maybe it's a first attempt globally of conducting a detailed LCA of tissue culture grown plants in avocado, comparing seed versus tissue culture. Where are the savings? What is happening when we are using glass, media, water? What is the environmental footprint? And this study will not only have impact on avocado, but this would help the tissue culture technologies for many other crops. So once again, a really beautiful thing happening, crossing the boundaries, delivering into that innovation space. So watch this space as Sudhir works more and publishes his first work on this area. And from avocado, we have progressed looking into Duboisia, which is a pharmaceutical crop. Turmeric has also gone into nursery trials, and looking at macadamias and more crops, just once again feeding into that pipeline. As I mentioned, this tool has also given us how can we conserve germplasm. And this is all about what we call frozen orchards. Having field repositories is fraught with dangers of pest and diseases coming in, cyclones, typhoons, whatever, hailstorms can destroy your valuable germplasm. There should be another way to conserve germplasm, and storing the germplasm under liquid nitrogen or cryopreservation is one such tool. Chris O'Brien has now delivered. We have avocado plants which were frozen using these techniques, and now they are growing in the field. So this is something to really, I would like to champion moving forward wherever it can happen, a cryobank or cryogermplasm bank, not only for avocado, but for other recalcitrant fruit, very much relevant to tropical and subtropical space. And we are also, as I mentioned, looking into gene editing technologies based on these tools that we have, whether it is avocado or it is strawberries, because this gives us the ability to hybridize germplasm without the need for flowering, and also use innovative tools of gene editing to deliver some desired traits, whether we want a dwarfing rootstock or a seedless avocado, or different unique colors or taste or flavors of strawberries, or disease resistance as a key feature. This work also has its benefits, across the boundaries. It has made us ever famous, as I say, because this was heralded as the technology, one of the 10 technologies at the Global Avocado Congress, which can significantly cut avocado tree wait times. Made me a little bit more famous as well with the cryopreservation work on the lighter side, guacamole on Mars, maybe that's a possibility. And of course, when you see on Twitter, rumors are that this belongs to Neena Mitter. So that's something, some of the perks of working on such crops. But coming to an end, our global leader, CHS really is poised to be a global leader in tropical horticulture. It is delivering research excellence, innovation, and impact for a thriving industry. It has very much in its ethos looking after the communities and the planet. And if you can see the team, that's the beautiful team. And I did take, smiling pictures of them. So they do smile most of the time, but that's the workforce that is delivering into this space as we are moving forward. And I'm very pleased to hand this beautiful center moving forward, because I'm sure it'll thrive even more in the coming years. And this is where I'm crossing borders again. So now I'm moving on, as Matthew mentioned, crossing the border from Queensland to New South Wales, to Charles Sturt University, and looking forward to staying connected with QAAFI, with UQ, and with other members, and widening and broadening those horizons. So just to end, as I was saying, borderless innovation, recognizing different pathways to success, guiding young minds to deliver for the future, and innovation that goes beyond discipline, organization, governments, industries, and geographies are really vital to saying hello tomorrow to the changing face of agriculture. So thank you very much, Matthew. Over to you.
Okay, thank you very much, Neena, and thank you for a truly borderless presentation there. You covered a lot of ground and a great summary of many of the outstanding developments in the team and of the team over the last 5 years, but also across your entire career at UQ, which I understand commenced 24 years ago. Is that correct? So a very long and embedded relationship with both UQ and with the Department of Agriculture and Fisheries. I don't see any questions in the Q&A yet, so I guess that gives me the prerogative of asking the first question. So I guess, Neena, I'm always struck by the advances that we've made in accelerating breeding in some of our crop species, and the challenge, but also the opportunity of applying those learnings into the more fragmented, necessarily fragmented horticultural sector. So if you had a crystal ball, how would you see that playing out over the next decade or so?
A good question, and this is where, Matthew, horticulture is a diverse sector. It is unlike any other sector. Even if you look at Queensland, the number of horticultural commodities or fruit and trees is large. So it is, we must acknowledge that, but then that is a challenge, but that's an opportunity as well. And this is where, as I was showing in some of the slides, there is a work going on across different crops and learning lessons from those different crops, but also building that knowledge then into looking at more fragmented crops, as you were mentioning as well, or industries which are smaller industries, whether it is custard apples or passion fruits, or even smaller than that, which are emerging industries or emerging horticultural crops. I believe the advancement in the sequencing tools, the cost of reduction that is happening in those tools, AI and machine learning enabling, and those prediction technologies, which the work is also going on in QAAFI in a full swing. Absolutely, I think because you gave me a crystal ball of 5 to 10 years, I think we would have solutions happening much faster as well. And just to give you an example, when we started working on avocado, Matthew, I was pained to see that even it was beaten even by Brussels sprouts. The genome of Brussels sprout was known, but avocado wasn't known till we published the full length genome of avocado. But now it is really, we can fast track that. So absolutely feeling positive that we would have that in our pocket sooner rather than later. But the industry needs to come together, and the effort needs to be an umbrella initiative. Otherwise, it just doesn't happen.
Thanks, Neena. And we have a couple of questions now. So one question is really about the role of AI in horticulture. And the specific question is about who are the top groups working on AI in horticulture, but perhaps more broadly, where do you see the place of AI in horticulture? You've talked about the digital twin area, but perhaps beyond that?
Oh, yeah. And as I was saying, AI is sort of, every time I'm looking at, even when I'm reviewing grant applications, there is an element of AI now in almost every aspect. It just depends upon, I always feel you need to you need to understand what's the challenge and then look at what the tool can deliver for you. So how do you use AI to answer that question rather than the other way around, sort of becomes much more productive. If you're thinking of AI in horticulture, once I mentioned some of the areas like in the digital twin space or DigiHort space, there is the use of drones and technologies. There is this huge push on robotics and automation. If you look at the work, some of the work going on at Wageningen University, now there are self-autonomous glass houses. Nobody enters that glass house. Everything, and I've seen that happening in the World Horti Centre as a display. The seeds are sown, the tomatoes are harvested, even the packing is done, and it goes out without even seeing a single human soul. They're completely self-autonomous. So those are some of the things that are happening in the AI space as well. But then also in terms of AI and machine learning, I mentioned about our crop protection or disease diagnostics work, a lot of imaging technologies and AI and machine learning technologies to scan large fields for pests and diseases, having that satellite imagery and maps available, not just for pests and diseases, but also maps on where the crop is being grown, how high it is, what it needs now. Those are also some of the solutions that can come through this AI and machine learning space. So it is just about how far you want to go and which boundaries do you want to cross by having AI as a tool.
Thanks, Neena. Charlie sends a question which is around farming methods like vertical farming. So how could we accelerate that in the future? And there's an example given here of perhaps alternating day-night cycles for improved growth. But perhaps more generally, just to get your view on where vertical farming or protected cropping sits in the landscape of our total horticultural production, say here in Queensland? What do you see it being a major component or staying as a niche opportunity, perhaps?
Now, protected cropping, the very word protected cropping, or in elsewhere in the globe, it's controlled environment agriculture.
Yep.
It starts at a polytunnel kind of a space as well. When you grow crops under nets or polytunnels, it is protected cropping. Then you come to the next phase, which is the glass houses, which can be semi-controlled, or greenhouses. And then at the end of the spectrum is the vertical farming, which is completely indoors. Each of these have their own benefits and have their own space, depending on the regions, depending on the climate, and depending on the crops that you're looking at and what is the value add you're looking at. In terms of Queensland, and we always used to say we have plenty of sunshine, plenty of good seasons. Why do we need to grow crops under protected cropping? But that is changing, and we all know that that is changing. Even if you think about breeding crops to address the climate change, but you don't know what the climate is going to be in 10 years' time. So this is where this value add of protected cropping comes in. If we are looking at controlled environment glass houses or the vertical farming spectrum of things, we can predict what's the climate going to be today, tomorrow, in 10 years, 20 years, 40 years, because that's what it is about controlled environment agriculture. And therefore, we can have the breeding done, the crops that can really grow well in there, and we can also look at that return on investment angle. But when you asked Matthew about vertical farming, I would like to add there, and there's a lot of debate if you look at the literature or even the LinkedIn or various posts, there's a lot of debate going on vertical farming, especially because a lot of companies came up initially and then there were some failures as well. But I always feel this is cyclical when a new thing comes into being, because sometimes there is a jumping the queue and putting it out there without understanding all the pros and cons. And maybe that happened at times with vertical farming, but absolutely brilliant space for us to work on. Not only once we can address those challenges of energy costs, have those pros and cons addressed, have the research done, vertical farming can really be a boon, can really diversify the horticultural sector. We should not look at it as a replacement of all the field crops, but it can add diversity and address those challenges. So a space to watch, and a space I'm sure and confident that vertical farming will grow once we address those challenges.
No, absolutely. A rich field for re-looking at how we produce crops in quite different environments. So a lot of interest for the future. I'm going to indulge myself by asking another question before we turn to the Q&A questions, but it's really about international collaboration, given your borderless theme. And it's always struck me that while there are international centres for research in field crops and animals, there is the World Vegetable Centre, but the fruit crops have really missed out. So in terms of the global effort to collaborate and provide opportunities for livelihood enhancement for smallholder farmers, but also nutritional enhancement, do you see through your collaborations with India and other nations in the third world that there's a way to perhaps plug this gap? It might be that the days of creating those kind of international centres is past, but there's certainly a need, I would propose. How do you see that opportunity into the future?
Oh, I concur with you. Couldn't agree more with you, Matthew, on that whatever be the terminology we use, whether it is centers or hubs or linking things, definitely we need that more and more to happen, especially to even manage the fast pace of technology development. If we are not internationally connected in answering those questions, we are losing that momentum. We need some, and I understand the question of, how do we bring that together? Some of those things are happening, as you mentioned as well. There is a bit of a footprint there, but it does need more, and also needs more connection with not only the developing world, but the developing or what I call some of the economies that can really push those boundaries and bringing it together. Philanthropy could play a big role as well, and industry championship could play a big role as well. We need to really think about how do we carve out those platforms where multiple industry partners, multiple international partners can actually come together. They can still have their own niche area to develop, but come together to answer some of the bigger questions or the bigger challenges that are facing, which are industry-wide, which will benefit the entire industry, but they can have their own niche area. I know that it can be done, has been done in some examples. UC Davis, I know, started as well a program of innovation in horticulture, which was started with the help of philanthropy and involved different partners. And the main focus of that was to deliver for sub-Saharan Africa and African nations. But then these are the kind of examples that we can grow. And as we were talking about AI, we can be all virtually connected and do few things together. But there is a huge opportunity, Matthew, to do that.
I suspect we'll be talking about that more into the future. So we do have a couple of more questions come in, only a couple of minutes left. Perhaps a quick one, which is really about the cost of bioclay, because it contains double-stranded RNA. I know you've done quite a bit of looking at this question. So would you like to comment on the cost questions? Yeah.
Thank you, Matthew, for asking me the favorite question, or whoever has asked this question. Thank you. Absolutely. When I started doing this work, research scale, we still buy RNA at $2,000 per gram. So it looks like, okay, can never have a real impact or a solution. But believe it or not, the cost of double-stranded dsRNA has now come down to even 50 cents per gram. That is the quote we are getting if you want to use it or buy it at large scale. And actually, Matthew Morrell did some calculations for us that if you have to spray 1 to 5 grams, I'm forgetting, of dsRNA for the entire wheat crop in Australia, you would actually need 65,000 tons of dsRNA. Whereas if you have to vaccinate the entire population or adult population of Australia, it may be just 800 milligrams. So there is the quantum difference. And that is what has led to the cost reduction. It always happens. It's the demand that makes the technology works. So yeah, it's quite possible. And as I mentioned, the first product has already come out. And for us, in terms of bioclay, the cost of LDH is actually in cents only. So looking good.
All right. Thanks. Another application of Moore's Law. I think we'll wrap it up there. So it remains for me to thank Neena very much for this eloquent presentation, to thank you once again on behalf of QAAFI for your service to QAAFI, its staff, our partners, and the university. And as we have reflected on a lot over the last few weeks, this is not a goodbye, but it is a transition to a new role, a new way of collaborating in a borderless world. So thank you very much, Neena.
Thank you. Thank you.